Hello again. My name is Laura Rodriguez. I'm from Mexico. I'm a scientist and artist. Now I'm working in ITMO University as coordinator of the educational program of BioArt. So I see some familiar faces. I think most of you already know what is BioArt. Yes, how can you describe BioArt?
Synergy between bio stuff.
BioStove? What is BioStove?
It can be different seed, right? So it can be soil, so plants, so you can see the jellyfish.
Yeah, it's actually a plant. So, yes, bio, life, right? And how the life is in Earth, like what is the minimum unit of life in the Earth?
Cell.
The cell. Okay. And in the cell, to be alive, we need to have the DNA, right? The NK. So everything from the NK to the level of the universe could be considered as bio or bio. So BioArt basically can include all of that. That's why it should be very silly just to narrow the single term of BioArt for a type of art that can include everything. Nevertheless, the popular term for making art with living matter is BioArt. Is it visible, the screen? Yep. Okay. So my interests mostly are in the connections between species and interspecies communication, but also empathy towards different levels of organisms, from insects to plants to humans. And in humans, my research interest is about mental health or different behavioral patterns. Okay, so for those that doesn't know what is BioArt, like my family, when I say that I'm a bioartist, they think that I'm painting with bacteria. However, well, no, this is not bioart. What we're seeing here is a copy of the Van Gogh painting, The Starry Night, made with some bacteria. So in these Petri dishes that you have 2 in your package, we can culture bacteria. And bacteria has different colors when it's growing by thousands, and we can see it, like these little dots or even big amount of them can look like paintings. However, there is a big difference between what is art and what is a craft. Crafting with biotechnologies will be something more close to this, using microbiology to create a craft that can be copied, that have an idea from another existing artwork, or that basically doesn't have a concept inside contemporary art. But bioart is part of contemporary art. When we are using the materials of biotechnologies like DNA modification, bacteria, plants, to express an idea that is based in the matter itself. So, if you want to express an idea about the chair, why should you use the fungi to make a chair? Maybe you need to make a connection between the fungi and the chair, and in that case it will become some sort of art object instead of a craft object. Don't misunderstand me, this is still quite difficult to do. If we will try to paint right now, probably our pieces will look like this. So it requires a talent. However, that's not the type of objects I will be talking during this lesson. You yesterday were with the professors of digital art and technological art. All of this is inside of Art and Science, the master program that we have in ITMO. And in generally, art and science is about making alliance between scientists, artists. But in bioart, it's also making alliance with other living organisms. So like the bacteria becomes your collaborator, or the plant becomes part of your team. And why we're talking now about art with biology? Well, art is quite fragile, right? And always reacts to what is happening in the world. And in the last century, we have the biotechnological revolution. In the 70s, Watson and Crick figured out the DNA structure. And then we have genetic modification organisms, genetic treatments in medicine. So it's affecting our life in this techno world. And that's why art also wanted a little bit a scoop of these new technologies. And artists start to work with this media. Okay, I want to make a pause here to understand a little bit about you. So please just tell me your name, what you do, and why are you interested in this workshop. Very fast. You did it yesterday twice, I'm not sure. Once, okay. But I'm always interested to know the person behind the desk, so please.
Hi, I'm Marina, and I'm an artist and designer, and usually I work with digital technologies. It's generative graphics, video, sound, and I am very interested in bioart, in bacteria, in microbes, in everything that's alive.
Yes.
Yes, and that's why I'm here now, because I'm looking for ways to combine my background, what I do now, and I'm trying to combine.
Okay, perfect, thank you. Next.
My name is Katya. I'm artist, painter, graphic, and I'm very bad speaker in English.
Don't worry. I remember we were together in a workshop in Moscow. No?
In last day, I understand that I can understand what
So you can?
Yes.
Great. Thank you, Kate.
Hi, I'm Maria. I'm an artist too. I mostly work with steel wire, and I make large-scale sculptures out of wire, and graphic stuff, and performance art, and I try to blend all those things together, and I think I just want more things to blend into that.
Awesome.
And more ways of making sense of the world.
Great. I like this last phrase. Thank you, Maria.
Hi, I'm Dasha. I'm actually, again, I will say that I'm sort of intruder here because I'm not an artist. I actually have management background and work in logistics, but I have started to study art history in very classic Academy of Raven art here in St. Pete. So I'm like studying classical art, but very interested in all these new forms that I imagine right now. So that's why I'm here.
Thank you. Thank you, Dasha.
Hello, my name is Nana, and I'm a linguist. My major is English, and my minor is Japanese.
Wow.
Now I'm teaching school children in a private school, and here I'm more of a science part. So I'm not an artist in classical understanding. I'm more of a scientist, and I want people to know science through art.
Okay, thank you.
Hello, my name is Alena, and my minor is languages also, and I used to be interested in biology and plants and bacteria, and now I want to come back to this hobby and to somehow connect it with art, and I'm really into this program and I wanted to enter.
Great. Awesome.
Hello, yes, my name is Irina. My background, I would say, more scientific. I'm psychologist and like scientific psychologist. And at the same time, I have a vast experience working with educational design programs and, well, for adult people. And recently I became very inspired by art and science approach on conceptualization level, how to approach things and different things, situations, yes. And right now I'm trying to be a leader of 2 projects, and one of them in bioart. So
Awesome.
thanks.
Great.
Hi, my name is Timur. My background is psychophysiology. I had some experience with science art. It was 2007. For a few years I was creating interactive neurophysiology sculptures with my friend back then. And after that I spent lots of time in education, in adult education, in business school, and now I'm trying to, in design of educational programs, and now I'm looking for a mean to
Great. Did you present the sculptures in some gallery?
Yes, even
Yes.
here in Erarta, one of them is that I did with Dmitry Kavarga. Back there.
Okay.
Together.
Okay, because you can also share later maybe the experience how difficult it is to present some living matter sometimes in the galleries, because well they were not ready sometimes to keep something alive there for a long time.
Even this, in galleries, they were trying to make me change my profession and become a technical person to support science art in galleries in Moscow. Because they said that this is a specific kind of an art that needs attention all the time.
True, 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 true. Yeah, I think even for a curator specialized in that will be good money. Yeah. Okay, thank you. Next one, please.
Hi, my name is Nikolai. I began to work as a lawyer in the sphere of intellectual property, accordingly to my education. But my life was connected from my childhood with tennis, and now I worked as a tennis coach for about 10 years. And my sphere of interest is human body, like a biomechanical system, and also I I'm interested in ethics questions and how we can enhance the body with something technology or biology methods, and is it good or not?
Awesome. Great, thank you, Nikolai. Next.
Hi.
Hi.
My name is Nast. I'm studying in the animation and graphic of computer games, and I'm really interested in animation and 3D modeling. And after some research of tendencies in contemporary art, I discovered Mo University and the Arts and Science specialty, and so after that I became really interested in it, so I want to practice some of the things that you are learning here.
Thank you. And?
My name is Olga. I'm working as a visualization. My background was architect, and I'm very interested in digital art, science, and computer art.
Okay, thank you. And everyone knows Ethan?
Yes.
Yes? No? So Ethan, 3 words about you.
3 words?
Yeah.
I feel the camera.
Film, art, and
Mexican. Yes, well, Ethan is my colleague and friend. He's also from Mexico, and as you know, digital artist.
I was lost. I was thinking.
Okay, so let's continue. So I was telling you about how the boom of biotechnology came or was very attractive to artists. So these are the discoverers of the DNA structure, Crick, and Watson. This is the first picture, the first selfie that we have of the DNA. But it was made by Rosalind Franklin. Do you know the story behind? Okay, so Rosalind Franklin in the 70s, she was working in science academia, but for example, she was not allowed to enter to the coffee room of the scientists because, well, there was not so many women in science in that moment. She developed a very strong personality to survive in this environment. And she had a collaborator, male collaborator, Wilkins. And Wilkins was making most of the contact with other scientists. And sometime in a party or something like this, Wilkins shared some of the results of Franklin to Watson and Crick and showed this picture. And Watson and Crick was like, this is what we needed to prove our motto. And then they used the same technology and published a paper, but Rosalind was not recognized as an author of this paper. She died, and after she died, the Nobel Prize committee recognized her as a contributor of DNA, but you cannot give a Nobel Prize post-mortem. So basically, there was like the story of the robbery of the DNA structure. So any of you see the DNA here? It's mostly like, you know, when you are pregnant and someone shows you some shadows and it's like, yeah, my baby, and you basically don't see anything. It's something like this, but it's like an X that is representing this part, okay? So scientists were able to understand that there was a double helix in the molecule. So if I give you a single concept of bioart, it will be very silly because it's still in making. But I like this concept created by an artist, Joe Davis, that he's working in Harvard, and George Church, that is a biotechnologist working with synthetic biology, trying to make DNA just from the basic structures and make a new organism. So they say that biotechnology is part of contemporary art that uses the biotechnology tools and methods to explore living systems. But these living systems are not objects, they are artistic subjects. Okay, that's a very important word when we are talking about bioart, subjectivity. And well, there are several topics that you can talk about with bioart projects, but most of them are focused in philosophical, societal, and environmental issues. Okay, however, there are other very good curators, for example, Jens Hauser, that say that there is no bioart. Or everything is bioart, or then there is no bioart. Because to create art, you need a living person, and the observer is also a bio. And what you are getting from an artwork itself, when you go and see a digital artwork from Ethan, you will get an impression. And that impression is what is making the artwork alive, and it's only working if you see it. So everything can be bioart, or nothing then is bioart. And also, this is not the only term that we use when there are artworks with living systems. In the 80s, when the boom of bioart started, it was also known as the name of transgenic art, living art, hybrid art, and last year appeared pan-bioart in China. That basically is what we know as bioart, doing art with living systems, but based in Eastern philosophies and trying to make a citizen science of it. So it's kind of a contra-movement of the Western movement of bioart that prompt or boost the use of living systems. So let's start for what is not bioart. There are some artworks made with fine art methodologies like painting of sculpture, that they are representing some of the philosophies or technologies of bioart. For example, transgenic organisms, this painting of Alex Strockman, when we have a pig that is growing livers and lungs to transplant to humans. This is a biotechnological speculative scenario. But this is not bioart, this is considered an artwork with biothematic. In that case, Dalí, making paintings about the DNA, it might be considered also as some artwork with biothematic. But then we have biomedia. Biomedia or biomediality is how I like to refer more often to this kind of artworks, not like bioart. But biomedia is when you take the living organisms and work with it or collaborate with them. The first organism that was presented in a gallery was the plant called Delphiniums in 1936, so around 90 years. So bioart is not that new anyway. Of course, this was presented in MoMA, but MoMA has only 7 years open, so it was not the big institution that we know nowadays. And the characteristic of these plants, they were artificially made in order to express more colors or to be taller. So it was the first organism artificially selected. I don't mean genetically, I mean you take a flower that has the characteristics that you like and the other the same and make them breathe. Okay. However, in 1928, we also have paintings with bacteria and fungi. Fleming, do you know who is Fleming? Yes, the discoverer of penicillium. So as a scientist, sometimes he was very messy and he was working with the bacteria. Then her lovely wife bring an orange and please eat, you haven't eaten in 3 days, you are just making research. And he forgot to eat the orange, then the orange has some fungi, there was penicillium, and he noticed that bacteria was not growing nearby. But he also made some paintings with this bacteria. Will you consider these as art? Yes?
The craft you spoke about?
Yeah, more like a craft.
Well, definitely we have the media.
We have biomedia for sure. Yep. So this is biomedia. There is not a connection between these, so there is not like a real concept. It cannot be considered such as bioart in contemporary art, but yeah, it's an artistic craft. Okay, let's go to the sophisticated part of bioart. You see here some faces, right? Do you know this project already? Only you or? Yeah, okay. So, the artist is Heather Dewey, and the artwork is called Stranger Visions. Heather was in the therapist, like many of us artists need to go to therapy. And she found a hair of someone else in the sofa. And she was wondering, like, who was this person that was here before me? How she looked like, what problems does she has. And then she realized that she had in her hand the DNA of someone else. So she could be able to make an analysis to identify who was this person. So she decided to go to the streets and collect some parts of garbage like the bonds from cigarettes. And well, there are a little pieces of your DNA in this kind of garbage. So she collected, extract the DNA, purify it. And then when you have the string of DNA, you can send it to big companies to receive the data. What kind of genes are in this DNA? And then make a comparison. There are big data banks available online where you can compare the DNA from your sample to another person. And she make a probability analysis to see how this person looked like. So in the DNA sample that she had, it said this person have 5 genes that look like similar to the blue color eyes. So she put blue color eyes in the 3D image of the person and created portraits of persons that she have never met. Okay, so can you imagine that you go to the gallery and find someone similar to you? Like maybe Heather was outside your house and collect some of your trash. Okay. So we will focus on what is biomediality, because in contemporary art, like the last 50 years, we have talked that the medium is the meshes. So what you are using for your artwork, the material and also the technologies, is already giving a meshes to the observer. It's not the same if you see a picture still that you see a TV moving, although it can be like the same image. But in bioart, let's say that the growth of the medium is the meshes. And it matters what are we using in order to express the meshes that we want, considering or rephrasing something that Donna Haraway says when we are trying to speak about other species. It's important that we need to understand how they are behaving, not from the human perspective, but from the species perspective. Okay, I want to make a very small intro about biology because I will talk about genes and DNA, so I just want like to have everyone an idea. We have the cell, and you have millions of cells in your body, right? And all of your cells have the same DNA. Do you know why the cell of your eye looks different from the cell of your skin? They have the same DNA. So they look different because although they have the same DNA, the eye is expressing this gene and this gene. But the cell that is in your skin is not expressing this, it's expressing this. Expressing, I mean, is making a protein from this. Okay, so in your eye, you will have the proteins and the structure to make your eye that are saved in your DNA. But it also has the instruction to make your epidermis from the skin. But all the instructions are in the same DNA. Yeah? So what it changed is the genes. And genes are these little parts of the DNA, fragments of the DNA that can be, they have the instructions to make proteins. And the proteins will make complex, and then tissues, and then organs, and then the organs will make systems like circulatory system, and then several systems like breathing system, circulatory system will make an organism, higher like human. But from here we also have plants and bacteria and fungi. So this is the main important. You have DNA with all the instructions. Little fragments can be translated into proteins. Okay? Okay, so I told you that everything from DNA to the level of universe can be considered bio. Okay? So let's talk about the first artwork of bioart in the modern world. It was created by Joe Davis. And Joe Davis, the artwork is called Microvenus. And it was the first synthetic DNA molecule that was inserted in a bacteria and then sent it to the space. What was this DNA message? It was a pictorial message. This image, that is the German rune that represents fertility or femininity of Earth, was translated into a binary code from computers, zeros and ones. You can see here still the same image of the fertility. And then something important that I forgot to tell about DNA. DNA is made by some nitrogen bases, some molecules, that is called adenine, thymine, cytosine, and guanine. And they always pair with cytosine, guanine, thymine, adenine. Okay, here. So this part of the DNA, let's put it like this, is TGCT, you see? And this other part is ACGA. And these are connected. So thymine, adenine, guanine, cytosine, cytosine, guanine, TA. So Joe Davis thought that one can be ACG and 0 can be TGC. So the 1 will be these 3, the 0, these 3, and you can make a string of DNA. He put inside the bacteria another DNA sequence. You see cytosine, thymine, so on. And this DNA was actually the text from the Bible, the Genesis beginning. he makes something similar than Joe Davis, but instead of using ones and zeros, he used Morse code. Morse code. And then translate it into DNA. Put it inside the bacteria, leave the bacteria have a happy life, reproduce, have several generations, then took the bacteria, extract the DNA, translate it back into Morse code, and then see that there was some mutations. For example, instead of say let men have the mind, it says let on to have the mind. So in this case, Eduardo Katz is collaborating with a bacteria to create the final artwork. The most important part of this, it was the first artwork when the author made a gene and a bacteria a subject. It gives you power to create something. So this was the first artwork when we are talking about a gene or a bacteria as a collaborator for the final piece. What do you see here? How it looks, what it looks like? Piece of skin, like a coat, piece of clothes. Correct. So, well, it is like a jacket or like a dress, but the author said there is a jacket, and it's called victimless leather. What you see here is a small bioreactor that has cells from a pig growth in the shape of a cloth. We use, or humans use, a lot of leather from different animals, and these animals suffer because they are deployed of her skin and protection. So Orton Katz proposes artwork saying like we could culture these cells without killing animals. So he just took a small piece of the cells of the skin of the pig and grow it in the bioreactor. This is how the final presentation looked like. So this growing dress, growing garment had the intention to confront ethically about the moral implications of wearing parts of animals. Okay? It was already very important because of the measures of how we are using other animals just for human benefit, but it became more polemic because when you visit the exhibition, it was still alive, so it was growing. And then people started to see it as a living thing. And when the exhibition finished, they wanted to kill it. Kill it. Can you kill just a bunch of cells? No? Yes. So it also brings questions about abortion, for example. If there is a bunch of cells, where life starts. So it became a very polemical artwork about the ethical rules that scientists and artists need to work to create these artworks. To be honest, in the laboratory, we kill cells every day. Okay? And it changed a lot. For example, I was working with cancer cells, and the scientists, one day came to the laboratory, and there was a cut in the electricity, so the fridge broke, and all the cancer cells died. And he was very sad because he didn't have cancer cells. I think it was the only option when someone is sad because doesn't have cancer cells. Usually, we're happy that we don't have cancer cells. So he continued working with growing structures with cells. He actually patented the methodology to grow these structures. And a lot of industry is trying to buy this patent in order that they can create the layers. But in order to grow these cells, you still need some nutrients. And these nutrients are getting from baby calves, like the fetus of the cows, in order to grow. So it's still not victimless. you are still using some other animals in order to grow these layers. So the artists do not want to sell the patent because he considers there is still ethically wrong. These little dolls, do you see the little doll here? Okay, they have inspiration in the Nicaraguan worry dolls. The mythology say that you buy it, you tell your concerns and fears, then you put it under your pillow, and when you sleep, this little doll will take care of your problems. So he decided to make it real, a living doll that you could tell your problems. So this is one example of how bioart is making fairy tales into truth, in some sense. And in Russia, we also have something kind of similar. This was for traveling, but I bought it during the pandemic, so it's not currently working. Okay, I will skip this one. I will go a little bit faster. This is one artwork that was awarded with the Golden Nika, I think is the pronunciation, that is inside the Art and Science Festival called Ars Electronica. That is one of the most important of art and science. And in 2019, it won the prize. Do you know this project? Okay, more or less. Yeah. So, Paul Benoist, the artist, was making a very simple questions, but very deep. How does exploitation or slavery smell like? So he was traveling to the mines to collect samples from the people that was working under slavery conditions. And all of you right now have millions of bacteria in your hands, in your skin, inside your body. And these bacteria are also helping you. Talking about psychology, there is some new trend about the gut-brain axis, that how the bacteria that live in your gut might influence, like the biochemistry that they are having, might influence also in the biochemistry of your brain. I don't know if...
So microbiome is extremely popular, and I have seen several art residences just about this topic.
True. True. So they are making, for example, implants from, it's called transfecal implants, when they change the bacteria from one subject to another in the hopes that will help, for example, with depression or with losing weight, because there are persons that, because of the bacteria that lives in their bodies, they are retaining liquids, for example. So it's very interesting. And bacteria also is responsible partially for your smell. So that's why we smell different. Paul Benoist took samples from the skin, isolate the bacteria from each person, and identify which one is more common in these conditions, and found three bacteria: Staphylococcus epidermis, Corynebacterium cerosis, Proponibacterium avidum. And he said, like, these bacteria that are always present, they are the responsible how slavery smell like, and create this wonderful exhibition when they have the three bacteria growing in bioreactors, connected the air by hose and tubes to a never used or a clean t-shirt that was being impregnated with the smell of slavery. There was a small hole near the t-shirt in order that the public could smell the slavery, so you will see people trying to smell how bacteria was impregnating the t-shirt. And I think that it's a multi-layer project. In the case that is exposing all the other microorganisms that live in you or that are part of you, and they are having some effect, but also linking the concept of slavery and the influence also, or the use also of these bacteria. Like now they are the slaves of Paul Benoist creating odor, right? Although he's kind of providing very good environment for them with food. I wanted to put back the video because, well, I like how it looks like. It's very techno-elegant, I will say. I don't know if some of you have worked in the laboratory, but the media, this liquid where bacteria grows, is not, I want to stop the video. It looks like this. It's like a very strong tea or a very awful coffee. It's really just like a dirty liquid. However, in the bioreactor, it looks so impressive. Why? Just because they put an orange light over. So this gives you the idea of something elegant, scientific elegancy. So this is very important when we are working with bacteria or living organisms, because they are usually repulsive for the majority of the persons. I think for artists or bioartists, we kind of get in love with the disgusting thing. Then we have Adam Brown with Irreverent Miracles on Demand. So maybe you have heard about these kind of miracles when a statue is crying blood. So he decided to investigate the phenomenon and found that this blood was actually bacteria. That when they are growing, they look like blood. They express a liquid that is very like sticky and red, that looks like blood. So he put in the installation the piece of bread and the bacteria, so the observer could see the bread crying blood, but actually was seeing the growing of the bacteria that is called Serratia marcensis. Okay. Then we started with DNA, then with bacteria. Let's go with plants. This is an autonomous organism. Doesn't depend on man to survive once it starts to work. It's from a Mexican artist called Gilberto Esparza, and it's called photosynthetic plants. In the nucleus, you have algae that is making photosynthesis, and it is connected to the columns that have bacteria in the soil, and the metabolism of the bacteria produce electricity. So by an anode and a cathode, the electrodes, it collects the electricity from the bacteria, turn on light, and the light will make possible that algae make photosynthesis. When the bacteria stops eats all the food and cannot grow anymore, the algae send more nutrients from its own nucleus, like more algae and water, so bacteria can keep growing and again make light, so algae can keep growing, and it's a cycle. So once it starts working, it can survive by itself, creating a dynamic of light and nutrients between each other. So it's an autonomous organism that was also presented around 10 years ago in Ars Electronica, also winner of this. I need to say that in Ars Electronica, the festival, bioart doesn't exist. It is called hybrid art. Okay. Okay, then we have bipolar flower. What is bipolarity? A way of life, yeah. So, well, it's presented, in very simple ways. We will have long periods of excitation and hype of emotions and long periods of depression, right? How a mental illness can be expressed in a plant. Does plant have mental illness?
I think no. I think no.
Yeah? I think we need to understand first if there is a mind in the plants. Plants have some system that allows the perception, right? We have plants like the sunflowers. They follow the light of the sun. So there is a system of perception. There is no eyes. There is no nervous system. So probably there is no pain as human feels pain. But there are some system of perception of how much humidity, how much light. So the idea of a mental illness for a plant, in human, a mental illness is a biochemical disbalance in the brain. So to create a mental illness for a plant, we need to create a disbalance in the biochemistry of the plant. So here we have, which one do you think have bipolarity?
The last one?
This?
Yes.
This one has bipolarity. Okay? What means bipolarity for this plant? They inserted proteins called zinc fingers. Don't put more attention to this, it's proteins. And this protein was pulling this gene to make more proteins. But they also inserted another type of protein that was trying to avoid to produce this. So it was a war inside the plant, trying to produce, not produce, try to produce, not produce. And that created that the plant had bigger leaves and more flowers. Of course, it can be prompt to degenerate if you have more cells, vegetal cells. Maybe you can have a tumor of cells, vegetal cells. However, in plants, a tumor of cells is not as dramatic as a tumor in humans. The author here is Adam Saretsky, and he couldn't present this in any gallery because it was a modified microorganism in America. And so what he decided was to make a performance, bring in the flowers for a concert. This is the scientist, and this is the artist scientist. And they are bringing the flowers into a chamber that makes music according to the living organisms inside. You see how the plants are in a container because they are modified organisms. If you bring it outside, it could have some extra contamination for other plants. And Saretsky is very ironical, as you can see. Okay, so he was making music for his bipolar flowers. The irony that Saretsky makes is between the overprotection in the science lab and the real life, making a comic anecdote of his experiments. Okay, so now from plants, let's move to humans. Marta de Menezes, she is a pioneer bioartist, one of the first female bioartists working in art and science. And one of her recent artworks is Anti-Marta. Now I will also use my body to show you. So in the 50s, there was experiments in America where they were using their own body to understand how transplant of skin looks like or works. So they were taking a piece from their skin here and put it here, and a piece from his skin there and put it here. Is your own skin, there will be a probability that your body take it back because there is no rejection. Nowadays we use this technology when there is some accident and someone get burned, so they can take part of your butts and put it in your cheeks. So Marta created a performance between she and her husband. They have been working together for more and living together like around 20 years. So she decided to make a statement of who are you and who I am. After 20 years of marriage, they kind of have been together so much that Marta had the need to create a performance to get her identity back. So both of them have the cut, and from Marta, one of her own skin come here, but this other came to Luis, and from Luis, one part stays with him, and the other came to Marta. So I don't remember which one it is, but let's imagine that this was from Marta. It will stay with her, and this one will be rejected because it have different type of DNA and cells. So it is a rejection of part of her husband, right? However, it's also very romantic poem to their relationship when they can be still together, but not be the same. Okay. It explores biological limits. I think is very sensitive and intimate artwork. I think also working with your own skin as a bioartist is very powerful. But the main idea was that, while we are together, we are still individuals. Okay? Working with your body can be also combining some sensors. A lot of people had inserted sensors and chips inside their body. The artist Moon Rivas created a performance that lasted years. She inserted a chip that is connected to the earthquake movements of the earth. So every time there was an earthquake, she felt a vibration. And she created performance and dances with it. Okay, I think it's also very powerful because an earthquake is not only the movement of the earth, it's also a lot of deaths, a lot of destruction and crisis in the world. So I will try to go a bit faster because now we are a bit late. We started like 20 minutes late. But let's go back to the collaborations. These larvae, Cadiz fly, normally they make a cocoon with the rocks that they found in the floor. But the artist put it in a container with gold and pearls. So they create a cocoon that is quite expensive, becomes the final artwork, and you can buy it if you have a few millions of dollars. Okay, so the larvae itself becomes a collaborator or creator of the artwork. Then we can go also to the environments. In bioart, we can also talk about the whole ecosystems. In this case, Robertina, she's talking about the ecosystem of the seas and the fact that the chemical industry is making in the seas. When you take, she focuses in antidepressants or in drugs for mental health, or any drug that you take. Like in the morning, you had a pain in your shoulder, you take a painkiller. Around 80% of the pill will enter in your biochemical digestion, but some other part will come in your pee or when you go to the toilet, and this will go to the sea. So there's our milligrams of drugs every day pour into the sea, and that can affect the growing of different organisms. So she was making a project collecting this type of data from the seas, and the final installation was these holograms where you could see different species, how they die or how they are modified by different drugs that humans are pouring into the sea. Okay, let's watch this video.
They may be created by humans for anthropocentric use, but as they will be creative and unpredictable, they might not necessarily stay the way they were originally intended. While the artistic values of the outcomes of this process are still in the eyes of the beholder, the questions regarding the possibilities are real. What will happen when such a system starts to express qualities?
Okay, I think I will better explain it meanwhile the video is there. You see some robotic arms. They are drawing. And they are drawing what the brain of a rat is seeing. But the brain of the rat is in a petri dish. It's not inside the rat. So the robotic arms were in Australia, and in a laboratory in USA, there was a petri dish with neurons of the rat. And the neurons have some electrical sensors connected to a camera. The camera was watching Maria, and sending some signals, electrical signals to the neurons. And the neurons were sending signals to the robot arm. So what the neurons of the rat was perceiving in a laboratory in USA was being drawn in Australia by the robotic arms. The main question of this artwork is, who is the artist? We have a rat artist, we have a robotic artist, or we have Guy Ben Ari as an artist. Of course, in the world of humans, the artist is Guy, and he is the one that sold the artwork and can get millions from his artworks. But also, the robots are selling the artwork. And we have here in ITMO, Ethan is working with a project when the robot is making also by his own drawings and selling it, right? So this project was also in Moscow, and the camera was watching the black square of Malevich and creating different representations. So this is considered, if you ask me, it can be also bioart, but it can also be technological art, right? Because we are using a living system, neurons. That's why in the beginning I told you that if I give you a single description of bioart, will be silly about me. For me, but for sure, for me, this is art. Do you think that it can be art?
But I'm very sad about mouse.
Mouse, yeah. Yeah.
I don't feel like it's art. It's very hard work for it.
Yes. Well, it probably was killed in order to get the neurons.
Maybe it doesn't want to make art.
Maybe, yeah. No, now it's like a slave, right? It's just making drawings. Okay, so that's a very good comment about the ethical issues to use other organisms. This was the first modified organism for art. Her name, or its name, is Alba. It's a bunny that was genetically modified to glow. They took a protein from a jellyfish and make it glow, right? But a lot of people was afraid of it. They thought it looked like a monster. What do you think about Alba?
I read that maybe it's fake.
Yes, true. They say, and from a scientific point of view, I mean, it looks very weird that even the eye is glowing, like everything is glowing.
And just only one picture.
And only one picture. Do you think that it matters if it is fake?
I think it's fake.
Yeah, I also could bet that it's fake, but I think that doesn't matter. The artwork still exists because he already put it in our mind. So there is an artist, Igor Kraft, that created a new color.
Yeah, speculation.
Yeah, so he claimed that he found a new color. And he created it, and a lot of companies are sending messages to him, companies from painting, like, can we buy your new color that you discovered? So on, but it's just speculation. So Eduardo Katz also made another artwork when he put hemoglobin, the gene from his hemoglobin of the blood, into a petunia. This petunia is called Edunia, Petunia and Eduardo. And then create these patterns with the blood. So a lot of people think that it's also like monstrosity. What do you think about this? I also consider that a lot of Eduardo's work is kind of trying to make an opposite direction. Like he presents something that can be scary in order to talk about the responsibility that scientists and artists have. So when people was watching the plant or Alba, they were scared, like this is a glowing monster, this is unnatural. So Eduardo came with a very good way to give back an answer to these people. And he asked them to put their hands together like this or like this. And you will start to feel some warmth if you put your hands like 1 centimeter far. This warmth is of course your temperature, but also a bit the infrared energy that your body is having in their skin. So we don't see infrared. Our eyes do not have the ability to see infrared, but some reptiles, some bats can see it. And for them, we are glowing monsters. Okay, so when we are talking about art and science, it's also about the perspective of the other. Of course, it's very difficult, and I think that it's not for everyone to try to be a plant or to try to be a bacteria, because the perspective can be also very difficult to understand. And then what is the difference between your fungi growing in your fridge? I hope you don't have fungi in your fridge, but if you have fungi in your fridge, what is the difference between that and an artwork in the gallery? Melissa Fisher, she created sculptures from her own body, took samples, and grew bacteria. So her final sculptures were growing like this, the fungi. And what is the difference between the fridge and this? Well, she's questioning herself, who I am. If I have one human cell for one bacteria living in my body, how important is this bacteria for who I am? That's why the artwork is called microbial me. So all the bacteria that live in myself. Okay, so this is one of the projects that I'm currently working. There is the removal of the cells from different tissues. You can remove the cells from different organs, like a chicken heart, but you can also remove the cells, please take it, and pass it, from different vegetal entities. In the back part there is a flower also, and this is a tail from flowers. Stem. You can be later free to watch it. And the advantage of this is that when we remove the vegetal cells, we're able to put back new cells. And these new cells can be cells from you. Okay? And we will have a layer of cells of human into the vegetal structure. This gave me the idea. And the important about the flowers and the leaves is that they have venation. So it can be connected to your circulatory system. Okay? Otherwise, it will die. But if there is blood coming to these cells, they might survive. So it gave me the idea of a speculative artwork where we have transplants that you can put in your skin, and it's called Becoming Plant. Becoming, for you, some of you that might be philosophers, was borrowed by Deleuze. But Deleuze mentions that becoming plant or becoming animal or becoming something is not about to resemble the plant or to act like a plant, but to create something new. In this speculative project, after you get your implant, you also get a serial of instructions of your new relationship with insects, for example. Humans relate with insects mostly like this. But plants relate with insects in different way. So once you get your implant, you have a new relationship with insects, with sun, with water, for example.